Good morning, everyone. I'd like to thank my mentor and guide, Dr. Kumar Prabhas, and entire organizing team of this conference to giving me this opportunity to present this topic. The NTRK fusion clinical efficacy and safety data updated things. So the NTRK molecule targeted Entrectinib, as we see in extensively in morning that many molecules like in BRAF, if we target only BRAF, there is a melanoma suppressing, but the risk of getting squamous skin will be higher. So similar way, there is a lot of research going on to develop a target which target the multiple pathways. So similarly here, it is this molecule which target the tyrosine kinase topomycin receptor kinase ABC, that is called as NTRK123. Along with it targets ROS ALK. So Entrectinib has shown clinical activity in patients with NTRK fusion protein as well as ROS fusion protein non-small cell lung cancer. So indications, NTRK fusion and ROS fusion. NTRK fusions, there are many solid tumors which it has expressions, and along with ROS. The dose in adult, it is 600 mg orally once daily. In pediatric dose, as it's 400, 500, and 600, depend upon the BSA. So disease overview, mechanism, action in a small. So basically, it is a gene fusions or specific type genomic alteration that derives oncogenesis. So if fusion frames, this can lead to expression of chimeric fusions, and if it is copy numbers, then somatic point mutations. So many other oncogenic kinase gene fusions, including NTRK and ROS, has been identified. So how it works, the chromosomal arrangement leading to NTRK123 ROS that causes fusion protein, ligand indenting, activation receptor, and signaling cascade, cellular effect, and then finally gives tumorigenesis. So tyrosine kinase are family of transmembrane TRK ABC, which encodes as NTRK gene 123. The tyrosine kinase proteins abundantly expressed in nervous systems, non-neuronal tissues, including cardiovascular, endocrine, and reproductive immune systems. TRK and ROS is dimerized phosphorylation on activations ligand building and gives the tumorigenesis in cancer. So basically, Entrectinib particularly binds to the ATP binding pocket, tyrosine kinase domains, act on TRK, A, B, C, as well as protein oncogene tyrosine kinase receptor ROS1, inactivating the downstream pathways and causing the clinical tumor response. So if we see, NTRK fusions has been identified more than 25 tumor types, including some with limited treatment options. So NTRK fusion disease has prevalence around 0.3% across all tumor types, which include brain cancer, mammary analogous secretory cancer of the salivary gland, secretory breast cancer, cholangio, colorectal, as well as head and neck, thyroid, lung cancers, and many more. And even in children, it's glioma, mesothelial neurofibroma, and infantile fibrosarcoma. So more than 80 NTRK123 fusion partners identified in a large number of tumor types. So basically, it is in rare cancer as well as in common cancer. In rare cancers, but it surprisingly has prevalence more than 90% in specifically mammary analog secretory carcinoma of thyroid, then breast with high prevalence tumors, and in rare, it is non-small cell, GIST, and thyroid. So current treatment guidelines for ROS1 fusion protein in advanced metastatic non-small cell lung cancer is first line is crizotinib and Entrectinib, and other recommendations is ceritinib, which is again as in ESMO guideline also comes as a first line indication. So basically, in NTRK fusion gene, there are 4 basic trials going on in phase 1 and phase 2 forms that I'll start RK1, 2, 3. Here I'm going to specifically discuss about the START 2 trial because it has a maximum number of patients for safety and efficacy analysis, almost 71% for NTRK efficacy, and 78 for ROS1 efficacy, and 332 for both combined safety profile. So START RK2 study design, it's global adult open label phase 2 basket trials, and it has included the NTRK positive, which TRK, ROS, and other things. So if we go clinical efficacy, If you give demographic data, most of the patients are white and Asian populations. Most of them received the first-line therapy, some in the form. Or CNS metastasis at baseline is around 25%, but both the reviewer. If we see the efficacy, if we see the complete response or partial response, it's more than 60% if we combine both in baseline or CNS metastasis. In no metastasis also, the efficacy is more than 60%, which we already previously discussed. Duration of response is significant with non-baseline CNS metastasis 13 months, with base metastasis also 6 months. Median PFS is 12 months and overall 13.7 months. If we see, as we see the number of patients in this study is sarcoma, non-small cell lung cancer, salivary gland, thyroid, breast cancers. So, durable intracranial response we are observing in NTRK patient with CNS metastasis is significant, almost 62%. ROS, I'll just keep it because next lecture we have on ROS about NTRK with considering the time restraint. Okay, if you see the ROS1 with the intracranial, as we discussed, there is a significant response, almost with measurable disease 78%, with 50% with measurable and non-measurable difference. So it is very good CNS penetrations and good efficacy. If we see the safety profile, the serious events is almost 40% in all, but if it is related, if you see, it is only the 9%. The related discontinuation is seen only in 5% patients, and dose reductions require in 25% patients only. So common adverse events, fatigue, constipation, dysgeusia, dizziness, diarrhea, nausea. So in summary, in integrated safety profile, Entrectinib was well tolerated. Majority of adverse event is grade 1, grade 2, non-serious. Most of them managed with dose interruptions. Treatment-related grade 3 events is only 32%. So it is well tolerated drugs, and it is grade 1 recommendations in NCCN guidelines for NTRK fusion. Thank you.